Hello, we're team number 5 and we are going to tell you and we are going to explain you a comparison between photosynthesis, cellular respiration, and fermentation processes. Our team is composed by me, Frida Patron, Jessica Robledo, Daniela Calvillo, Raúl Ramírez, José Eduardo Morales, y Eduardo Camarillo. Okay, first I'm gonna to explain you about the steps of photosynthesis. Well, the simplest step or the simplest explanation of the steps of photosynthesis is that the carbon dioxide, that is H2O, turns into oxygen, that is O2. But what is the process? Well, the process is made in the chloroplast, and it's the carbon dioxide enters in the plant in the thylakoids, and then with the help of the light, it turns to the oxygen. But first, it passes through a process by which the ATP and the NADPH turns into ADP plus P and ADP plus with the help of the Calvin cycle that it's carried out in the stroma. But the Calvin cycle is carried out in 3 steps, that is filtration, reduction, generation, and regeneration. In the filtration, the CO2 enters in the stroma, and then it turns that the NADPH turns into NADP plus, and also the ATP into ADP in the reduction. Then the glucose comes out, that it's the same as sugar, and the regeneration also takes the ATP to the ADP. So that's the steps of photosynthesis best explained.
So now we'll talk about the enzymes and proteins involved in the same process of photosynthesis. First, we have here the general equation for the photosynthesis, that is 6CO2 plus 6H20. Then with the help of sunlight, the process is helped and is converted to C6H12O6 plus 6O2 plus the carbon dioxide, water, and energy from the light. So we have 2 reactions. First, the light reactions, and then is the Calvin cycle. In the light reaction, it's happening in the thylakoids disc, and in the chloroplast of plant cells, there are a series of enzymes and other proteins called photosystems 1 and 2. And in the Calvin cycle, the ATP and NADPH are used to reduce or fix carbon dioxide to produce glucose. Here we can see the different formulas for each of them. And at the end, the energy required for releasing cleavage of high energy is 18 ATPs and 12 NADPHs.
Now we're going to talk about the steps of cellular respiration. These consist of 3 steps or procedures that are the glycolysis, the Krebs cycle, and the electron transport chain. First, we need to know what is glycolysis. Glycolysis is the pathway that glucose catabolism performs. This is carried out in the cytoplasm, and also we are including here the decarboxylation of the reaction of pyruvate. Then we have the step number 2 is the Krebs cycle, which is carried out in the mitochondria, as well as the decarboxylation of pyruvate. In here, the acetyl will be used in the Krebs cycle, and in which most of the reducing equivalents they will be used by the electron transport chain are generated. The final step is the electron transport chain, which is carried out in the mitochondria. So as we said, in the Krebs cycle, we create or we generate the reducing equivalents, So this will help us to speed up the electron transport chain. And as a final result, we will get H2O. This through the consumption of oxygen and the gain generated during the transfer of electrons as a result of the synthesis of ATP.
Then we have the enzyme and proteins involved. As previously stated in the general question, glucose and oxygens are transformed with the cellular respiration into CO2 plus water plus ATP. First, we have the glycolysis in here. Glucose, ATP, NAD, and the coenzyme A are transformed into acetyl-CoA plus NAD plus CO2. The glucose is transformed into a pyruvate, which will be used in the Krebs cycle, where the NAD transforms into NADH with the help of the coenzyme. Why? Because it's an electron carrier. In the glycolysis and also in the Krebs cycle, 2 ATP molecules are produced, but it's in the third step in the electron transport chain where the acetyl, by the use of a ATP synthesis, is transformed into ATP, and the results of the electrons are received by oxygen, which is the cause of the H2O to produce, while 34 ATP. So in a net value, 38 ATP molecules are produced from this whole process.
Steps of fermentation. There are two types of fermentation, the lactic and the alcoholic. In the lactic fermentation, the glucose must be absorbed by the cell to start its metabolism. The route by which glucose is catabolized is glycolysis, and if we have oxygen, the process will be cellular respiration, but if there is no oxygen available, the final outcome will be lactate. Alcoholic fermentation. In the first step, a carboxyl group is removed from pyruvate and released in as carbon dioxide, producing a 2-carbon molecule called acetate. In the second step, NADH passes its electrons to acetaldehyde, regenerating NAD+ and forming ethanol.
Enzymes and proteins involved in fermentation. Well, as you said there are two types of fermentation, lactic and alcoholic. And one difference between those is that lactic takes place inside the human body. The net energy in both of fermentations is 2 ATP molecules. The fermentation of this class are purely chemical reactions in which the enzyme acts as a simple catalytic agent. Enzymes are protein in this chemical reaction. Enzymes break down starch into simple sugars that are usable by yeast. This is to form pyruvate molecules, which is also known as glycolysis. Yeast feeds on sugar and stretches.
That was all. Thanks for watching. Hope you enjoyed.